What up? What up? What up? Hi, ladies and gentlemen, the books is in. The books are in. Yep. They're in. They're here. What up, everybody? Good morning. ETM Hotel, Rennie, Shawn. Welcome in peace. My name is Shawn. I'd like to thank everybody for tuning in to episode 7 of Science with Shawn. Back in the fair to all the Netaru that will be in the building. Yeah. And the Netarettes who will be in the building. Yeah, we're going to get it in today, man. You know what I'm saying? I know it was a late night last night for a lot of people, and they probably ain't even got up yet, but, you know, sleep is for the weak, they say. So I'm back with another episode of Science with Shawn, you feel me? Before I get started, I want you to like the show, share the show, tell a friend, subscribe to the channel. If you're not subscribed, you know, click the subscribe button, click that bell, and get the notification when we go live. We kicking science over here. So there's a few things that we're going to deal with today. And before we kick that off, man, if you know, for everybody that's watching the stream live, I want you to go ahead and go in the description of the video, and I want you to grab the information that's in there, right? Instead of people speculating and assuming and not knowing everything, I want y'all to know everything. So I want y'all to go and grab the PDF docs that I've put in the description of this video. All right, one of them is Vaccines and Related Biological Product Advisory Committee meeting, December 10th, 2020. It's an FDA briefing document on the Pfizer-BioNTech COVID-19 vaccine. The other one is the development and licensure of vaccines to prevent COVID-19, guidance for industry. The other one is the Pfizer-BioNTech COVID-19 vaccine, Vaccines and Related Biological Product Advisory Committee briefing document meeting the same day, but it is the 492. So there's one that is 53, and then there's one that is 92 pages. Then I want you to get the extra guidance for industry, general principles for the development of vaccines to protect against global infectious diseases. That's 13 pages. And last but not least, I have a doc directly from the FDA Gov website regarding COVID-19 vaccines. FDA has rigorous scientific and regulatory processes in place to facilitate development and ensure the safety, effectiveness, and quality of COVID-19 vaccines. So I want you guys to jump on, grab that information, like the show, share the show, tell a friend. Tell a friend. We're about to go all the way in. You know, get this out. Chavis, where you at? Represent one time. Share it on Facebook. You know what I'm saying? Let's get the people up, man. Look, we gotta wake the people up, yo. It's our job, our duty, our responsibility. Let's get these people up, man. You know what I'm saying? So while I am let me make my little post. Make my little post. You know what I'm saying? Let me get my science involved. And then we gonna get it crack-a-lacking. So I hope y'all had something to eat. You know what I'm saying? I hope you had your breakfast this morning. Shout out to Sister Juju. Today is Juju's birthday. Make sure y'all, you know what I'm saying? Make sure y'all tell the sister happy birthday today. Let me see who earned. Let me see who in the track. All right, okay. Chavis in the building. Zane in the building. Ramon in the building. Peace. Yeah. Hotel to the nature rule. And where the naturettes at? You know what I'm saying? They sleeping on us, yo. Sleeping on us. Sleeping on us, yo. Just give me a few minutes, man. I am working on it right now. And we're gonna get it cracking, yo. Yeah. We're gonna get it cracking. How y'all doing this morning, man? What y'all think about last night's show? I know it's a little delay, so you know what I'm saying? I don't know if everybody caught up with all that. It was 2 shows. There was a lot of information in those 2 shows. Some contention and all that in 2 shows. So what y'all think about that, yo? It's cool. Yeah. About to make some breakfast while I'm a show on. Yeah, man. Get your breakfast, yo. Get your breakfast, yo. Straight up. How many people woke up and watched Power? You know what I'm saying? Y'all watch our Power episode this week. Well, none of that foolery in there, that coloring in there this week. You know what I'm saying? Y'all only got one more week of that anyway before the show end. But I know how people are, yo. Yeah. Also, man, look, support the merch store, man. You know, the link to the merch store is in the chat. The link to all of our books is in the chat. You know what I'm saying? It's the holiday season, yo. You want to give people some information, something they can hang on to. I guarantee you, Spiritual Combat and Killmonger's Corner Volume 2 will provide you with some real substance to help educate and teach your kids, yo. We doing all these sign shows. You know what I'm saying? FYI, they got those trucks ready. Those trucks going out. They putting the vaccines on trucks and in the airplanes, yo. The trigger being pulled, yo. They pulling the trigger. We need to know what's up. So I'm giving y'all, you know, firsthand information from the FDA, and we're gonna get into our bill. Y'all know how we rock it. You know what I'm saying? I'm just trying to get a couple more people in here, and we're gonna rock out. You know, we're gonna rock out. All right, so there she go, yo. Y'all could tell her now. She in the building. You know what I'm saying? They go to birthday girl. She in the building. Peace to the Naturettes. You know what I mean? Olokun. Peace to you, Black African Power. You know what I'm saying? Yeah, man. We need some people in here, yo. Let's go. Before we get it crack-a-lacking. Yeah. Hopefully, yeah, I see people sleep, yo. I don't think, I mean, I understand. You know what I'm saying? People want to chill out Sunday morning. They don't expect Shawn to pop up out the blue with some stuff. They expect maybe this afternoon or evening, you know what I'm saying, when everything get to cracking, you know, nah, I'm doing a morning show, Sunday morning show. Yeah, I went to sleep late, up early. Straight like that. Sleep late, up early. That's how we doing it. That's how we doing around here. We sleep late and we up early. Straight up. All right, so let's get it cracking, man. You know what I'm saying? Anybody to catch the stream after the fact, hopefully you'll like the stream, share the stream. But we got a good build today. I think y'all are gonna appreciate some of the information that you hear. And you know, we're gonna go from there. Now I know my brother Dave Lee. Shout out to brother Dave Lee. Shout out to my brother Rob. Peace to you, Rob in the chat. You know what I'm saying? Let me see who else. I got another Rob. Let me get it. Let me do this right. Let me do this right. Let me get Rob Rich involved. Let me get this brother involved. I gotta get my people involved, yo. Hold up. Let me drop this link in the back. I don't want nobody crying, yo. We ain't gonna see Kofi. He don't show up on my shows. You know what I'm saying? I'm throwing shade this morning. He don't show up in my shows, yo. But he better show up to, you know what I'm saying, everything else. Let me see here. All right, let's go. Share my screen. Let's get it cracking.
This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is twive, This Week in Virology, episode 693, recorded on December 11, 2020. I'm Vincent Raccaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's 50 Fahrenheit, 10 Celsius, clouding over, supposed to rain tomorrow. And fortunately, the stump grinder that was running in the neighbor's yard is now all done with the job. But I just wanted to mention that because longtime listeners will appreciate the stump grinder arc.
So you didn't want to have a stump grinder as our background music.
That would have been some distracting background noise, yes.
Also joining us from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It is 54 Fahrenheit, 12 Celsius, and sunny here. Big change from Wednesday when we talked.
Yeah, it snowed a little, right?
It did.
It's all gone. And from Austin, Texas, Rich Condit.
Hi, everybody. We've got, what is it here? Sorry. 67. Overcast. Drizzly. And to Brienne, a lot different than it was when we talked.
A lot different than it was. And from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. It's the same here as it was when Brienne spoke. Whatever she said.
The Aaron Diamond AIDS Research Center at Columbia University Irving Medical Center, that's where I am, is seeking a high biocontainment laboratory assistant manager to help run the day-to-day operations of the center's biosafety level 3 core facility and to assist investigators in SARS-CoV-2 experiments. Applicants should have practical experience in performing cell culture microbiological techniques and basic molecular biology. Applicants must have a bachelor's degree in science with 3 years experience working in a BSL-3 or 3 years of non-BSL-3 experience and a willingness to travel for BSL-3 training.
Hey, they speaking to you, Africans. They speaking to you. They got a job opening. Who's ever qualified should apply.
Yeah, I'll put a link in the show notes. You can apply to that. We're very good at recruiting people for various positions. The other Institutional Biosafety Committee position, not committee, but you know what I mean.
Yeah.
Institutional Bios. environmental health and sciences. Let's put
The
it that way.
biosafety job, right?
Biosafety job, and also Amy, a technician for Amy. We recruited both twive listeners.
Oh, you got one. Good. Great.
And Amy's tech has come from Seattle. Welcome.
My goodness.
She's a
Great.
twive listener, so maybe she's listening, or probably Amy's keeping her too busy now. So there'll be a link for the show notes for this BSL-3 position. And I think, Dixon, you could apply for that. You've got cell culture experience, right?
I do. I've cultured all my cells, in fact.
A little bit of news. We have 2 papers quite interesting for you today. A little bit of news first. As everyone must know, the FDA Advisory Committee yesterday voted in favor, 17-4, right, for the FDA to make an EUA for the Pfizer vaccine?
17 to 4 to 1. One abstained.
One abstained? Like what's the abstention about? I don't get that.
Maybe they felt they had a conflict of interest.
I think all of the no's as well as the abstention were about the 16 to 17-year-olds,
Yeah.
as opposed to it starting at 18.
So abstention is a way of saying, I don't want to say no, but I object to this part of it, maybe, right? Anyway, it's good enough for the FDA to take note. And in fact, they're saying they're going to approve it tonight by the end of Friday. And so next week, the vaccine might be rolling out, which is cool. It's the first phase here in the US of getting something going.
Here.
I'm very excited. I think it's good stuff. And
I'm not sure where I stand on the 16 to 18-year-olds, because the issue is they weren't in the trials, but the reasoning is, well, like close enough.
there were a few people in trials, very few, apparently.
Yeah.
And not enough.
Yeah,
That was the objection. The 4 people said there are not enough kids to really make a decision.
Yeah, and they're. I know Moderna is starting right now. They are starting a trial on, I think, 12 to 17-year-olds. So the expectation is that this is going to get tested, and that obviously, since it's already been tested and proven effective in adults, you're not as concerned. And then you go into kids. That's why you don't start in kids, because it's a worse place to have a problem. But I think the expectation is that, sometime in the spring, we'll get an answer on efficacy in kids.
Yeah, so this is only an emergency use anyway, right? This is
Yes.
So there's another date for the actual approval for everybody. So does anybody know whether in England they raised the same issue before they approved it?
The issue of what? The young age?
So yeah, 16 to 18-year-olds, yeah. Because
I don't know if they approved it for that group.
well, that's right. I don't know any details of that. I thought.
Yeah,
I'm not really sure.
No, because they did the same. They also gave an emergency approval or the equivalent of it. And I don't know the details.
Pfizer said it will apply for full approval in April after they've collected 6 months of safety data. And then they could sell it to anyone who wants it. Right. But right now, emergency use. And so as we've talked about, healthcare workers, elderly people, et cetera, first. And by the way, speaking of getting in line, the Times has a vaccine timeline. You can go to it and put your age, your occupation, comorbidities, and it will tell you how many people are ahead of you in line. And I am 180 million behind.
I think Brienne's probably going to win this, but I've got 268.7 million in front of me.
Yeah, I'm putting it in right now.
Yeah.
It's pretty funny.
My wife, it says, it doesn't even get a number. It just says very few.
She's a healthcare worker, yeah.
Worker.
Very good. So she should be able to get it in the next couple of weeks.
She should be the first one in the household vaccinated. My daughter is 15, so she won't get it till they approve it for kids.
Yeah. That's very cool. Dixon, you ought to put your numbers in, Dixon, because...
Oh, look how far back I am in this line.
How many?
You're behind me.
268.7 million.
Oh, we're in the same group. I see. Okay.
Did you look at the picture at the bottom?
Yes, it goes
There's
on.
actually a line that shows people and where you are in that line, and I have to scroll quite a while.
Yes.
It's very funny. Dixon must be higher up.
All right, watch this. Just so y'all kind of know, I don't think that she puts it on the screen, but there's a data bank that she goes to. You see her down here. She's pulling up something where she actually can see where she gonna fall in the whole thing as far as overall. Like, we way back in the back. There's so many frontline workers, so many elderly, so many pre-existings and people that are, that's going on is ridiculous. And we ain't paying attention to none of that information, yo. We ignoring everything else that everybody else in the scientific community and the rest of the world are paying attention to, where they are, what's going on with this thing. And we're arguing over minute things that was already resolved 6 months ago, 9 months ago, a year ago.
Because you're so ancient.
He must
Dixon.
be. I don't want to reveal yet what's happening here, but okay. I just.
And Condit is going to be more or less like me.
I'm 118.5 million.
Oh, he's ahead of me. Cool. Maybe it
I'm
goes
older.
by state. Do you put your state? Oh, you're older.
You
That's
put
true.
your county.
Put
Yeah.
your county.
Put your county.
I'm only behind 7 million others in New Jersey.
Oh, that's not bad actually. It only
I'm
takes
only
you
behind
6 months.
5.4 million in Massachusetts.
This is very fun. It's a lot of fun. Amusing. I'm just going to stay in here and podcast for the next year, and I won't worry. But what is this setback of the Sanofi Glaxo? Does someone have?
Oh yeah, so I popped that in there. Let me get back to the notes here. So Sanofi Glaxo, which I'm really disappointed about this because I was looking forward to this one. It's a subunit vaccine. Uses the same technology as one of Sanofi's flu vaccines. And if you didn't know, Sanofi is the world's leading supplier of flu vaccines. So if you got the flu shot, as you should every year, you have probably gotten a Sanofi shot. I know I did this year. They and Glaxo have been collaborating on developing this. They were planning to start a phase 3 trial this month. And being Sanofi, since they operate at such a massive scale, they were expecting to produce 100 million doses of vaccine by the end of this year. So this month. And they were going to make 1 billion doses in 2021. This is a vaccine that can be stored at refrigerator temperatures.
Wow.
So this is kind of, vaccinate the world type stuff, both in scale and in storage. The problem is that they had, stop me if this sounds familiar, they had a dosing miscalculation in their phase 1/2 trial. And of course, this also happened with another trial.
Yep.
So some people ended up getting a lower dose than they should have because there was an error in measuring the dose that they were supposed to get. The adults, ages 18 to 49, demonstrated a fine immune response based on that, but the folks over 60 had a weaker immune response, weaker than people who'd recovered from COVID-19. So then they're going to back up. I think they're doing a phase 2B or something just to back up and redo that, make sure everything's good. They're expecting to start their phase 3 then next year, and now they're looking at late 2021. The interesting twist here for approval, if everything goes well. The interesting twist here, though, is they're saying now, since this has slid so much, obviously we're going to have Pfizer, Moderna, possibly AstraZeneca, maybe somebody else on the market by then. So then who's going to want this? Right. I mean, we're piling up the vaccines. Well, but who's going to want the fifth one on the market that, comes out in November of next year after everybody's already gotten their shots from other companies? And while there will, I think, still be a sizable portion of the world's population that'll need it, the interesting angle that Sanofi is taking on this is, well, how about if we do a comparative trial? So they are deliberately setting themselves a higher bar and saying, instead of doing a placebo control, because the other issue is, how are you going to start a phase 3 trial in an environment where people have access to a working vaccine?
Yes. Yeah, you can't get rid of standard of care, right?
I mean, I'm very pro-science and I want to see these trials work, but there's no way I would volunteer for a trial that might give me a placebo when I can go and get the real shot against this virus.
It's called
all right. This is important information here. Since they fell behind in the vaccine race, and then you have 3 front runners, potentially 4 by January, maybe even 5. Well, you take away that one, so you're just going to say 4. You got 4. So now that you got 4, the only thing the 5th or 6th or 7th person can do is to do a unilateral trial, a comparative trial. It would only make sense. What can we learn from a comparative trial? Johnson & Johnson up against Moderna, up against Pfizer, up against AstraZeneca, up against Sanofi. When all of them are comparative, we can see, based on a phase 3 trial of that nature, if they got 40 or 50,000 people, we can see how long the immunity lasts in every last one of those vaccines. We also can compare the adverse effects, right? The adverse effects of those vaccines. I'm saying that to say that since it's so many millions of people that have to be vaccinated, this vaccination process is going to take a while. And we're sitting here waiting. Those of us who are way back in the back, the 13 to 18-year-old trials will be out, and we'll know more about that before a lot of us become vaccinated. This kills the myth of the guinea pig. So while we're in waiting, we're collecting more data and we're getting more knowledge about the things that are beneficial to us and what we can improve upon to make an informed decision. But when we put these companies against one another in a trial phase, in a phase 3 trial, it will uncover which vaccine may work best for us. Now, mind you, some people still have allergies. They got to resolve the issue for allergies for certain people, right? They're telling people with certain type of allergies not to get vaccinated right now. Let them work through some things. So that helps them go back in the lab and deal with the molecular structure for people with allergies or a certain type of allergies to fix that problem. This allows us Black people to look at science from a different window and say, hey, based on all of this information, I can stay away from Moderna. I can stay away from Pfizer. I may need to stay away from Johnson & Johnson. I may need to stay away from AstraZeneca. Sanofi may be the vaccine for me. Why? Because out of all 5 tests, Sanofi had the lowest adverse effects. Its immunity is lasting longer than the other 4. The other 4, we've learned that immunity was only lasting 6 months, or it was only lasting a whole year. But after these phase 3 trials and we've come out of it, Sanofi is lasting, 2 years, or it's still going with the immunity. You get things like that. Like we get to decide, right? Now, most people like, I ain't getting no vaccine. I don't care, and none of that, right? Okay, don't get the vaccine. That's you. We talking to the people who are trying to learn more about the vaccines so that they can make an informed decision and then actually deal with it from a scientific perspective and be more aware. I'm not telling anyone to go get the vaccine. What I'm trying to do is to give you more information, more resources to help you make an informed decision so that you could do what you need to do at the time that you need to do it. Let's get back to the show.
Standard of care, right?
Yeah, so standard of care, the idea is to take people and randomize them into either one of the approved vaccines or Sanofi's vaccine.
Yeah.
And then see how that works out. And they would have to either match or beat those vaccines, which would be cool, and so I appreciate them doing that, and I hope this ends up working out.
So I understand they were the sixth company engaged by Operation Warp Speed,
I think
right?
there's 6. I lose track.
Yeah, I think so. Okay.
So Vincent, my number is 283K.
Wow, pretty good.
283,000. Okay.
Yep. So
Very
I can
good.
see the end of that line. I can see the end of the line from my house.
What is it, New Jersey? I bet you're pretty high up in New Jersey because that's US
That was
280.
New Jersey. That was New Jersey.
All right, so pretty soon you'll be able to come in here, Dixon. Let me ask you a question, Dixon.
Yeah, please do.
Here's a question. Once you get your vaccine, can you throw your masks away?
Absolutely not.
Y'all heard that. Once you get the vaccine, should you throw your mask away? Absolutely not. We know that. Why do we know that? Because they're propagating that information for an important reason, right? They don't know how this thing is going to work with other people. You have shedding that takes place, which could expose other people, right? Now, it may not be affecting you, but you still may be shedding. Now, I put this out the other day, but if you don't have this, this is the actual published journal regarding the actual vaccine trials, right? This is one of the many. Good job, Brother Shawn. We get it. All right, so let's deal.
Why would I want to do that? I can still, let's see, what can I do with my mask? I can show people that I still care whether they get infected or not.
I'm just using this opportunity to look at your fish. That's all. Sorry.
Oh, all right.
Okay.
I can show you more pictures if you want.
Yeah, another time. We saw them at the end once of some.
We did.
Thing. Actually,
Yeah,
speaking of
that's
vaccines,
true.
one of our papers is about vaccines, and in fact, we were talking about the AstraZeneca Chadox one vaccine last Friday, and people asked about data, and they said, "Well, it hasn't been published yet." And then after twive on Friday, it was published. And
Right.
Kathy pointed it out on Wednesday, so I thought we'd take a look at it. It's published in Lancet: Safety and Efficacy of Chadox COVID-19 Vaccine AZD1222 against SARS-CoV-2, an interim analysis of 4 randomized controlled trials in Brazil, South Africa, and the UK. And of course, we know the results already. This is the one where some people got a half dose first time and so forth. We know the numbers, but I want to go over a few things that are quite interesting and you might not have seen before. And of course, this is a very big author list collaboration between University of Oxford and AstraZeneca and many other places who've participated in the clinical trial. And the reason I want to do this is in particular to take a look at the side effects, okay? Because they have an appendix document here that has all the side effects, and we'll get to that. So this was done in 3 countries, right? UK, Brazil, and South Africa. And they pooled all the data to get at their final efficacy numbers. And of course, they had this one group where they got, everybody got 2 doses, but one particular group, the first dose, which was made by a contract manufacturing organization, right? By somebody else, in other words, they used spectrophotometry to quantify the amount of virus, and it turned out to be different from PCR quantification. So they ended up packaging half the dose. That's what happened, right? And
this reminds me of trying to get an accurate read on my solutions of RNA and DNA, okay, which absorbs at 260. And you got to be careful that you don't have anything in there other than RNA and DNA that might absorb at the same wavelength. In particular, a reagent that's used commonly in our procedures, beta-mercaptoethanol, that absorbs very strongly, okay, and can give you a false high reading. So you think you've got a lot when you don't have as much. And they talked about in this particular process, they didn't discover until later on in the quality control that there was an excipient, had to look it up, Okay, that is something, some other reagent, probably innocuous. Okay, but that absorbed at whatever wavelength they were doing their spectrophotometry, and gave them an artificially high reading. Hey, got to be careful.
And well, this is what happened with, as far as I can tell, with the Sanofi situation. It may even be the same thing for all I know. And it raises the important point that this is a new virus, right? And we are accelerating it's being done as safely as possible, but some of the steps in the protocol, little things like this can suddenly say, "Oh, wait, we didn't check that, did we?" So,
well, if you spent I've kind of shadowed my ex postdoc Susan DaCosta as she moves through biotech, okay, and learns what this is all about, and encounters the rigor of standard operating procedures and quality control and all the rest of it. And it's a discipline and a skill all by itself. And this just goes to show you how important that kind of rigor is.
Yeah. And they caught it.
Yeah, they caught it.
I mean, they got partway into this trial, and they had given some people initial doses, and it's not a large number of people who got this low dose. It was for a short period of time that they were dosing while they were checking, triple checking everything, and said, "Oh, wait a second," and then fixed it. But as a result, they've got this subgroup that got low first dose, higher second dose, and it actually turns out to be kind of interesting, as people have probably already heard.
And if I remember correctly, the way they caught it was that the people who were getting a lower dose did not have as exaggerated an initial reaction to the shot, and some alert physicians said, or people running the trial said, "That's funny. These people aren't reacting the same way as the other people. I wonder what's going on."
And so the people who got this were in the UK and in South Africa, not in Brazil, as we originally said.
Right. And I also thought there was an interesting part in the description of the protocol where they talk a little bit about the fact that sometimes they were not able to do a perfect 4-week later boost. And so they also spent a little bit of time thinking about, how long after the first dose was the second dose given. And I think that that's going to be important sort of practically as we move forward, because that may be what happens with some people in real life.
All right. So we have the Lancet study. You have the link in the chat. If you just joined the show and you don't see the link in the chat, let me know. I can repost the link to the actual study. Basically, what they're going over is for the next 20 or 30 minutes, well, 28 minutes is the actual study. They're talking about an instance where you had two countries randomly given a smaller dosage, first dosage, and then a bigger second dosage. Although although they all got double shots, it's telling you right here, the UK, study design of participants, continuing single blind phase, half clinical trial in 5 sites in the UK. When the trials will begin, April 23rd, 2020. Enrolled 1,077 healthy volunteers at the age of 18 through 55 years and previously described. Briefly healthy adult participants were enrolled after the screening include the pre-existing health conditions. Participants were randomly assigned to a 1-to-1 to receive the COVID-19 dose. The viral particle standard dose measured using the spectral. Let's see what this word means so y'all know exactly, so we can be on the money. This word means it's spectrophotometer. So now, the apparatus for measuring the intensity of light in a part of the spectrum, especially as transmitted or emitted by particular substances. All right, big word there. And then, or they say this word here. Look that up. One thing about the laptop you like, while you're reading an article, that's all you got to do. All right, meningococcus. We need Anka up here, Kakasi, to pronounce these words. We had some fun with that. A bacterium involved in some forms of meningitis and cerebral spinal infection. All right. So we continue on with this. There's a group A, C, W, and Y. Conjugate vaccine as control. As an open label, non-randomized subgroup of 10 participants were given 2 doses, 28 days apart. So, roughly they're saying around 3 weeks, 3 to 4 weeks. The study was originally planned as a single dose study of 88 participants. In the phase 1 part of the study remains recipients of a single dose. However, the protocol was modified to a 2-dose regimen following an amendment on July the 30th. For the remaining phase 2 cohorts, as a result of the robust booster response identified in evaluation of the earlier immunogenicity, cohorts with the booster dose given at the earliest possible time. Continues to go on. The COVE-002 is a continuous single-blind phase 2 out of 3 study in the UK, began on May. So what we're doing when we're regarding the UK is they're telling us exactly when the phase 1 started, phase 2 started, how many people were part of the phase 2, what was going on with the phase 2, and then we move on. So we got the COVE-002. Now we're at the COVE-003. COVE-003 is regarding Brazil. So we get all the data out of Brazil that should link up to the UK and South Africa. South Africa, they listed it as COVE-005. They tell us the earlier part of their studies when it started, how many people was involved, and how many of the small group. Okay, I'm kind of trying to summarize it here, but I'm gonna let them get back into the bill because I know y'all don't want to really hear my voice. Y'all want to hear the experts.
And in fact, some of the issues they had with the second dose are things that we may very well see because they had a supply issue, I gather, at one point, and it delayed people's doses by a couple of months. So there ends up being a much bigger gap than was originally planned for some of these patients.
Pfizer has already said that it doesn't really matter much if you get your second shot second week or third week. So there's a lot of wiggle room in the second dose, apparently.
There was enough concern about this, or interest, shall we say, in this that in the paper they wound up splitting up the different boost times or intervals, okay, and comparing the results in each of those groups. And one of the things I found amusing was that they had a way of saying in the paper, in this case and in others, that we're presenting this because the reviewers asked us to.
Yes.
Yes.
Nice. So the as I said, UK and South Africa, some people in those arms got the half dose. Then a couple of other things I found very interesting. So first of all, they immunize people with vaccine or with placebo. Then what do? The participants were asked to contact the study site if they experienced specific symptoms associated with COVID-19. And they got regular reminders to do so because you can imagine you might not remember. Those who met symptomatic criteria had a clinical assessment, a swab taken for some kind of nucleic acid test, and then blood samples, right? So if you have cough, shortness of breath, anosmia, or ageusia, you would have, which is loss of smell and taste, right? You would take a swab, and then they would see if you're SARS-CoV-2 positive because they want to confirm it by after the symptoms, right? So that's how this works. And then they say, we're counting the people with confirmed COVID-19 and the vaccine and the placebo. Although at the time that the data are collected, of course, you don't know who's who. You just know there's persons in the trial. And then later when it's looked at by the Data Safety and Monitoring Board, they get to see who's who. Right.
And so they also did screening for asymptomatic infections.
Yes.
Which I appreciated a great deal.
So they were asked to provide weekly self-administered nose and throat swabs from week 1 after the first vaccination. They were provided kits for this. And this is very interesting. We're going to look at the results from that, which are quite interesting. So this was done, it was not done in Brazil, they say. So
it wasn't everybody who had it.
Let me see. What were the 4 sites again? UK, Brazil, South Africa.
South Africa.
Just 3 sites. Excuse me? 2 in the UK.
2 were in the UK.
Because there were 4 trials, right? 2 of them were phase 1, 2. 2 of them were phase 3. And they pooled all 4 for the safety data, which is perfectly legit. And for the efficacy data, just the 2 phase 3 trials where they assessed efficacy. So there's different numbers in those different assessments.
The other thing they did with the efficacy trial to part of the trial to keep the data clean, you want to cast as broad a net as possible to find any problems, which is why you look at the safety data on pooled. And then the other subdivision they made in the efficacy is if in the analysis, if they were seropositive for SARS-CoV-2 at baseline before getting the shot, so they'd been infected before, they could still go in the trial and they could still get the vaccine because you want that. But they were excluded from the efficacy results.
All right, so real quick, participants across all 4 trials were asked to contact the study site if they experienced specific symptoms associated with COVID-19 and receive regular reminders to do so. Those who met symptomatic criteria had a clinical assessment, a swab taken for a nucleic acid amplification test, and blood samples taken for safety and immunogenicity. In the UK and Brazil, the list of qualifying symptoms for swabbing included one of the following: fever of at least 37.8 degrees Celsius, cough, shortness of breath, and nausea or agazia in South Africa. The list of qualifying symptoms for swabbing was broader and additionally included myalgia, chills, sore throat, headache, nausea, congestion, diarrhea, runny nose, fatigue, nausea, vomiting, and loss of appetite. Now, in all the studies, if participants were tested outside of the trial, either in their workplace, if the healthcare worker or private providers, these results were recorded and assessed by a masked independent endpoint review committee. The source of each swab was recorded, plus the details of the test kit were available. So they're telling us exactly what they were doing to these people, when, where, and why, how. To test for asymptomatic infections, participants in the Cove 002 location were asked to provide a weekly self-administered nose and throat swab for the NAT testing for week 1 after first vaccination using kits provided by the UK Department of Health and Social Care. Participants were given home kits provided by the DHSC that included step-by-step instructions on how to do a self-swab and linked to a demonstration video. The site trial team provided support for logistics of the packing and returned test kits and tracking swab results to participants if required. Swabs were taken by participants in their homes and posted to dedicated DHSC testing laboratories for processing. Participants were directly informed of their results by text or email from the National Health Services. Swab results from participants in England and Wales were provided to the trial statistician on a daily basis by the NHS and matched the individuals based on personal identification, name, date of birth, NHS number, and postcard. Swab results from participants in Scotland were unavailable to study team at the time of the data cutoff for this analysis, but will be included in the future analysis. Any swab results that were not able to be matched to a study participant using at least 2 pieces of personal data were not added to the study database. All right, now they're telling us clear in Brazil there was no testing plan for asymptomatic infection. In South Africa, asymptomatic infection were detected from swabs obtained by the study attended, but are not summarized here as there were only a small number of time points for detection of those cases. Now, all cases of COVID-19 were reviewed by 2 members of a mass independent clinical review team who will assess clinical details, including medical history, symptoms, adverse events, and swab results, and assign severity scores according to the WHO clinical progression scale. Now, for asymptomatic participants in the 002 in the UK, weekly swabbing continued both before and after participants reported symptoms to the study site. Thus, a participant who reported symptoms and was clinically assessed might also have additional swabs return positive results through an asymptomatic testing process for several weeks. In addition, due to the large number of healthcare workers enrolled in these studies, some participants were, according to their workplace, testing policies, and these results were also entered into the database for review mass endpoint evaluation committee. Further exploratory assessment of the length of the time participants remain net positive, and the sources of the information used for the case detention will be done in future analysis. We're caught up.
So if you were in the trial and you got the vaccine and you'd previously showed antibodies to SARS-CoV-2, then your efficacy to it, that's nice for you, but it doesn't count for the trial, which is an important point because that could really skew your results otherwise.
Yep. And the other thing that they do that we should just point out here, so I listened to the FDA meeting yesterday, and one of the things that came up in some of the discussions were that the placebo group was getting saline. And so they assumed that people were able to identify whether they were in the placebo group or the vaccine group, and that perhaps that changed their behavior. In this trial, instead of using sort of saline as their placebo, they used another vaccine. They used a meningitis vaccine. And so if you were listening to that yesterday and heard about the concerns about people self-identifying as placebo based on reaction to the vaccine, that would not be an issue here. Both groups could have had a reaction to their shots. It was just a question of whether they're SARS-CoV-2 or meningitis.
And there's another reason why that's important, and I kind of prefer this trial design. There's evidence that's been kicked around and that we've even talked about on twive that maybe just kind of generically goosing the immune system with something like BCG or polio vaccine or whatever provides some level of protection. And so you'd hate to put all this money and effort into developing a very specific SARS-CoV-2 vaccine and find out that everybody could have just gone and gotten their flu shots and it would have worked, right? So it's nice that they've got a real vaccine as the control in this trial. And who knows, that might even explain why they see 70% efficacy versus the other trial seeing 95. We may never know if that was.
Well, what you can say is that this meningitis vaccine doesn't protect you because the placebo had most of the COVID cases.
It did, but maybe they would have had even more if the placebo was saline.
Could be.
Is the point.
Could be. Well, we could compare it to the mRNA vaccines where they have used just saline, right?
Exactly.
Numbers are similar, right? 90%. 23,848 participants all told. And a couple of things that I wanted to point out. Well, first of all, the standard two dose, we've given these numbers before, 62.1% efficacy. And then the high dose regimen, 90% efficacy. Here, a small portion of the participants were seropositive at baseline. And that's interesting. People have often asked us, if you're seropositive or been infected, are you excluded? And apparently not. They just enrolled everyone because I think they don't do the serology until you're after enrolled and already got the shots. So this is looked at in retrospect, and I'm not sure it made any difference, right?
Well, and you also want those people in the trial because a lot of folks have had it, don't know it, will go get the shot. And you'd really like to know if you have some issue or, is it a good or a bad thing to get the shot if you've already had the virus? And in this case, it seems like probably fine to go get the shot.
138 people in the UK and 235 in Brazil were seropositive, at baseline. Yeah. I don't know what happened to them. I'm sure we could find it in the data.
But 3 participants seropositive at baseline had a subsequent PCR positive swab.
So that means they got infected.
They got infected. One got 3 weeks after the first dose. The other two other participants in the control group had symptomatic infections 8 weeks and 21 weeks after the baseline sample was taken.
So they weren't protected, basically, right? They were not protected.
It's another example of the issue that natural infection does not seem to be a reliable way to have immunity against this virus.
It's quite interesting. There's only a few out.
I heard that natural infection in no way to gain immunity against this virus. Natural infection in no way. Let's just hear him say it again.
infection does not.
They don't do the serology until you're after enrolled and already got the shots. So this is looked at in retrospect, and I'm not sure it made any difference, right?
Well, and you also want those people in the trial because a lot of folks have had it, don't know it, will go get the shot. And you'd really like to know if you have some issue or, is it a good or a bad thing to get the shot if you've already had the virus? And in this case, it seems like probably fine to go get the shot.
138 people in the UK and 235 in Brazil were seropositive, at baseline. Yeah. I don't know what happened to them. I'm sure we could find it in the data. But
3 participants seropositive at baseline had a subsequent PCR positive swab.
So that means they got infected.
They got infected. One got 3 weeks after the first dose. The other two other participants in the control group had symptomatic infections 8 weeks and 21 weeks after the baseline sample was taken.
So they weren't protected, basically, right? They were not protected.
It's another example of the issue that natural infection does not seem to be a reliable way to have immunity against this virus.
Natural infection in no way seems to have immunity against this virus. So you remember that they kept on saying, let's just everybody get infected. One day we'll just get rid of that. You just heard this man tell you straight up and down, this is proof positive that you're not going to gain natural immunity with this virus. So you're going to dismiss that part. That needs to be considered here. What's the risk of recontraction? Don't look, you're going to get it. Reinfection is in the air. Not only that, but if you're not gaining immunity against it, what is COVID-19 doing to your body once it re-enters a second time? It's going to further its damage. We went through that last night on the second show, Pseudo Killers. I talked about how the virus creeps into your body and how it mutates and attacks. And we need to know this stuff because it's important. But you just heard an expert say that this is proof that you can't gain immunity from this virus. Natural immunity. Let me put the natural in front of it.
Quite interesting. It was only a few out of the hundreds.
It was only a few, yeah.
Yeah,
I mean, and across the board, we're talking about small case numbers, and I think the big one, this low dose, standard dose, the people who got the low dose first. There's a lot being said about, well, it was 90% effective in that group. Well, yeah, but you're talking about 3 people in the vaccine versus 30 people in the control group got the virus, and the confidence interval on that is really wide, and this is a tiny number of cases, and so there's some concern that maybe even with that, it's not quite right.
So for all the people who in some corners of the internet are really worried about vaccines potentially leading to enhanced disease if you've already been infected, this is certainly not, all of the data you would want on that, but this is some data to indicate that the individuals in this trial who were in that situation were okay.
Yeah. And I think we have similar findings from the Moderna and Pfizer trials, don't we? Or did they exclude seropositive people?
I don't know. Good question. I would have to go back because those have been published, right?
Yeah, I think they included them, but
actually they haven't been published. I'm sorry, right? The Moderna
they have not been published.
Phase 3 have not yet.
But one of a major concern with the enhanced disease issue is that if you get vaccinated and then get a natural infection, might you get worse disease?
Yeah.
And I think that if that were happening at any largish frequency, we would know it by now.
Yeah, and given the size of these trials and all 3 of these trials taken together, that's just not showing up.
It could be 1 in a million, though, right?
It could be 1 in a million, and that's why The full approval for Pfizer, is waiting for 6 months of safety data because they want to roll it out for emergency use and see how things go and monitor it all along.
All right, so that answers the question about everybody else receiving a vaccine, right? You remember they were like, so they're gonna start making everybody get the vaccine. That is further from the truth. Right now, they're spending the next 6 months, collecting this particular data before they ask or request for full approval. We heard that earlier in the show. So we have at least 6 months, and you heard Sosa say this last night. We have at least 6 months worth of data that we're going to have. Really, we're going to have 6 months of data as of when people start getting the first response and all of that. But if we're talking about the UK trials, we're talking about the same 6 months. So we're talking about like May, right? We're talking about May when Pfizer's going to ask for full approval. So we have that data that'll be collected to us in May. Then we have data from 3 months prior to the information that we just got published today. So we go back those 3 months, and then we go back, the earlier trials that actually took place in the phase 1 trials. And then we look at the results from the phase 1, because people are forgetting. We're so focused on the phase 3 that we ain't talking about the phase 1. So we still got data from phase 1 that we're going to be able to observe. We're going to have data from phase 2 that we're still going to be able to observe. We're going to have data from phase 3 that we're going to be able to observe. So truthfully, for me, it's looking more like we're going to have a year's worth of data that we're going to be able to observe that's going to benefit our sound judgment and our decision making in this whole entire process. All the while, Sanofi around that time is trying to do a clinical trial regarding comparing all the vaccines. Remember that. Don't lose that point. Teddy, you said something about rapid testing for the COVID. If it's available to you to do the rapid testing, you know what I mean? You want to know the method is there. There's evidence behind it that it's accurate. The testing has gotten better than it did initially in the first few instances of testing. So the swabbing is, you know what I mean? You still got to get swabbed up, but they're just testing for antibodies. So the antibody testing. So this is why you see athletes, testing 3 or 4 times a day. If they come in the facilities, they got to get tested. If they leave the facilities and come back, they got to get tested. If they go outside and exposed to something and come back, they got to get tested. They go eat lunch or take a break somewhere, they got to get tested. You know what I mean? It's a continuum of testing, and these results are showing to prove that they are pretty much valid. So, yeah. So we continue to do all of those things and just kind of progress forward. Let's keep building.
So let's talk about these asymptomatic infections, right? They were asymptomatic or unreported systems in 69 participants. Okay, so then they could calculate the vaccine efficacy with the idea that this is a small number, right? In the 24 low-dose, standard dose recipients was 58.9%. In other words, how good was it at preventing an asymptomatic infection? Whereas it was 3.8% in the 45 participants who got standard dose. Both not great, right?
Right. So it seems like this is a vaccine that's preventing disease more than preventing infection, but it can prevent infection in some group of people.
This is why after Dixon gets vaccinated, he's going to have to continue wearing his mask.
A mask.
Exactly. Yes, because
they did find in this group, in the folks who got the virus, tested positive after they'd been vaccinated, they didn't get very sick. In fact, I think none of them were hospitalized, isn't it? Versus a number of hospitalizations in the placebo group.
Anyway, it's interesting.
So it does seem to be very protective against what
serious
you're really
disease.
afraid of, which
That's
is getting
right.
very sick or dying.
Good enough for me.
You can still probably transmit, and so
You could
you're
still
gonna,
probably carry it, and that's what we've been.
Until a good number, a fraction, 70 to 80%, Peter Hotez said last night, he said, we did those numbers here in Houston, 70 to 80% of the population is immune. Then you might be able to, then that would inhibit transmission sufficiently, because
And that would have to be sterilizing immunity.
No, it doesn't have to be. But the number of people that now protected. Oh, I see what you mean, because they're still going to be infected.
Yeah.
So yeah,
it
because
doesn't matter.
typically
You could vaccinate if you had a vaccine that was purely protective.
That's true.
And not sterilizing. You could vaccinate 100% of the population, and then one person comes along who's not vaccinated, they could get the virus and die.
Yeah, because those 100% are still shedding. You're saying, right?
Those 100% could still be spreading it like crazy.
Well,
and probably would be. They'd be safe.
According to the numbers here. If you do the low dose, high dose, what is it, 58%? So that leaves quite a large percentage that is not protected and still going to be shedding.
You can't get to herd immunity with this if those numbers hold in the larger population.
Yeah, that's right. It's very interesting. So what's the outcome of this? We have to wear masks forever?
No, the outcome of this is you're going to have to vaccinate everybody. And if people are not vaccinated, they're going to run the risk of dying of this virus.
Yeah. And everyone means as far over 90% as you can get, right?
Yeah, it means everyone. It means, like, how many people do we have in the world now? 7 billion? Pushing 8 billion?
7.8 billion, actually.
7.8.
It's a lot of doses. Wow.
It's a lot of doses.
Yeah. It also means we might need some of the things like distancing and things like that as longer term changes.
Yes.
Okay. Yep.
Hey, are y'all understanding what they are saying about this? Like, are you like really comprehending that until, now let's just say the United States, like, if we just look at our population per se, and we vaccinate 90% of that, we run a risk. Here's what we run a risk of: travel. We travel outside the United States, where the vaccination rates aren't high or will be high, or there's not enough people vaccinated. That means you could run the risk of traveling outside the United States and running into an area possibly where these people, man, are a struggle. So we have to continue to wear our mask, practice our distance, keep our distance, and just be safe, man. Wash your hands, sanitize, all that. Just be extra safe. We have our reasons. And you heard old boy say it earlier, man, people run the risk of death, right? And run a risk of death. But I dropped some information from the actual findings that most people love to talk about. And that had to do with the deaths, right? So let's look at it real quick. Let's start here. All right. Yeah. Across all 4 studies, the vaccines had good safety profile with serious adverse events and adverse events of special interest balanced across the study arms. Serious adverse events occurred in 168 participants, 79 of whom received the ChAdOx1 nCoV-19, and 89 of whom received the MenACWY or saline control, which is the placebo. There was 175 events, 84 in the ChAdOx1 and nCoV-19 group and 91 in the control group, 3 of which were considered possibly related to either experimental or controlled vaccine. a case of hemolytic anemia in a control group in the UK phase 1.5 study occurring 10 days after the men WY vaccine was considered possibly related to intervention and has been previously described. A case of the transverse meningitis was reported 14 days after the booster vaccine is being possibly related to the vaccine, with the independent neurological committee considering the most likely diagnosis of the idiopathic short segment spinal cord demyelination. A potentially vaccine-related serious adverse event was reported 2 days after vaccination in South Africa in an individual who recorded a fever higher than 40 degrees Celsius, but who recovered rapidly without an alternative diagnosis and was not admitted to the hospital. The participants remains masked to group allocations, continued in the trial, and received the second dose of the allocated vaccine without a similar reaction. See, they don't tell you all of that. That's why you got to read, yo. That's why you got to read. Now, there were 2 additional cases of transverse meningitis that were originally reported as potentially related, but later determined to be unlikely to be related to the vaccine by an independent committee of neurological experts. All right. So remember earlier they had said it was related. Now they double back with the evidence from an independent committee saying this was not the case. One case that occurred 10 days after the first vaccination was initially assessed as possibly related, but later considered unlikely to be related to the site investigator when further investigation revealed pre-existing conditions with this person, but previously unrecognized multiple sclerosis. All right. The second case was reported 68 days after men ACWY vaccination. While considered possibly related to the site investigator at the time of reporting, an independent panel of neurological experts considered this to be unlikely. All trial participants have recovered. I repeat, all trial participants have recovered or are in stable and improving conditions. All right. There were 4 non-COVID-19 deaths reported across the studies, 3 in the control arm and 1 in the COVID-19 arm. All right. That were all considered unrelated to the vaccine. The cause of death assessed as road traffic accident, blunt force trauma, homicide, dang, somebody was just killed, and fungal pneumonia. All right. Unrelated to the vaccine. Unrelated to the vaccine. Unfreaking related to the vaccine. So with the memes and all of the paranoia, you know, that's what's going on. So again, I gave you this already. Let's see how much they want to talk about. They talk about this a little bit more the next 9 minutes. And then they get into the oral antiviral blocks, which is key as well. And I'm gonna go ahead and throw this in the chat so we can get ahead of the curve. And I'm gonna let them build for a minute, and then we can get into this.
Another thing I wanted to discuss, well, first of all, there's an appendix, 2 appendices, and one of them, table S4, are the baseline characteristics: age, sex, BMI, ethnicity, percent health and social care workers, comorbidities I wanted to point out. So the 3 comorbidities that are represented: cardiovascular disease, respiratory disease, and diabetes, right? So those are people at risk for serious disease, and they want to see if the vaccine is okay in them, and it is, right? That's important. So that's only those 3, but that's important. The other is the side effects, because a lot of people are concerned about side effects. And what they do is they have a huge table where they have serious adverse events, because, you know, the moderate ones: fever, pain at the injection site, fatigue. You know, we talked about this on Wednesday, you know, a third of the control people are fatigued because people are just tired. And all those things are not game-changing side effects, but the serious ones, right? Those are the ones. And there's a big table because, you know, as they said, some people died during this trial from a traffic accident, blunt force trauma, someone hit them over the head, or homicide, or fungal pneumonia, right?
Probably not caused by the vaccine.
not caused by the vaccine, but when you immunize, 30,000 people, or you put them in a trial, things are going to happen to them. It's
right,
the
and
way
you have to report all of that.
Yeah.
And this table, S6, serious adverse events by organ class, very granular, and it goes on for 6 pages. But what they do is divide them into the events in the vaccine group and the events in the control group. So you go down and you, oh my gosh, 5 cardiac disorders. Yeah, there were 6 in the control group too. And you go down all those gastrointestinal disorders, same thing in both groups. Injury, poisoning, procedural complications like animal bites. There was one in the control group. I guess the vaccine prevents against animal bites. Bone fractures, other infections. What else? Metabolism disorders, cancers. 4 in the vaccine group and 5 in the control group. The point is, when you hear about side effects in the vaccine group, you have to look at these charts in the control group, because people tend to leave that out. I heard the other day someone say, there was Bell's palsy in this trial and 3 people in the vaccine group. Yeah, but there were 3 in the control group too.
Right.
So that's the key. And most people, I think, are not going to go through this table and look at it. But in every case, the serious events happen in both the vaccine and that's why, and the control group. And that's why they say they're not related to vaccination. Homicide, yeah.
All right, team. All right. Look, so check this out. Yesterday, Uncle was talking about the Bell palsy because there's a meme that was going around social media. And you just heard this man say that the Bell's palsy happened in 3 people that was in the control group. Well, let's say the placebo group. And there was 3 people that was a part of the actual vaccinated group. So there was a total of basically about 6 people who had Bell's palsy on both sides of this, right? So they're trying to insinuate that the vaccine causes Bell's palsy, but it doesn't. Why it doesn't? Because there was 3 in the control group that actually got the placebo that actually had it too. But you don't hear anything about them people. You know what I'm saying? So we should find that very, 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 very interesting. Hold up. Let's cook. Cause Angie, she sometimes she get a little off, right? You know what I'm saying? Like we really don't agree with it right there. So Angie, what do you actually imply when you say vaccines cannot be one size fits all? If vaccines are affected disproportionately, so are you saying that we Black people, we genetically we have a different genetic makeup than white people or Asians? Is that what you're saying? I'm just asking for clarity. Now you may catch this later, but there's a little small delay, but I'm trying to figure it out. So we know that Bell's palsy happened in the controlled in 2 experiments. All right, the placebo and with the vaccine. So what does that say? Do we wash them away? You know what I'm saying? We went through this yesterday with Unc. You know what I mean? So I think that we really should. Wow. We really should pay attention to those things and kind of deal with it like that. But I'm down here dealing with the tables with the vaccine efficacy. And you know what I'm saying? I'm looking at the charts and the data. Now, remember, in the description, I put all of this in here in the description because we really ain't got to listen to the scientists, or we could listen to the scientists, but we also can follow along because we got what they got. This is what they reading. They looked at the Lancet, yeah. They dealing with the Lancet, but here go the primary. Here go the primary right here. You know what I'm saying? So this is how we deal with it from that perspective. But I'm gonna let them build because I said I'm gonna let them build.
There was 1 homicide in the control group. So all the things that happen to people, here they are, and they're in both groups. So that's why they're saying so far after 2 months, this thing has a good safety profile, right?
Yeah. And by the time most of us who are 200 million people behind in the line get this vaccine, we'll have a lot more safety
information.
data and a much bigger.
That's right. Yes.
But even if you're at the front of the line, I think there's good reason with this and with the other two vaccines. I feel pretty confident in them, at least for protecting against disease.
Now, go ahead, Rich.
I just wanted to point out something that I've been thinking about in these data and get your reactions to it. My bottom line is that I want to see more data.
Sure.
Because, for example,
Before what? You get immunized or just in general?
I want to see more people sick. Okay. I'll give you an example. In the low dose, standard dose thing where we're saying the efficacy is 90%.
Right.
The total number of people who experienced disease was 33.
Yes.
In the trials that we've been evaluating so far, they've been saying that their cutoff for being able to get a statistically significant number is somewhere over 100.
Yeah.
150, sometimes on the order of 200. And here we're talking about 33 cases. So based on the standard that's been set for the other vaccines that I've seen, and I'm no statistician, but that is lower than what would have been an acceptable number. So in order to, it's been widely touted that this low dose, standard dose protocol gives 90% efficacy. I want to see more cases.
Yeah, right? That's why I called that out. Yeah, that's why I called that out earlier. And they're pretty forthright about that in the paper.
Absolutely. Nobody's trying to hide anything.
Right. No, this is a small number. The one thing they didn't mention in the paper was that, the details of how this has been hyped. But I think the researchers themselves are very, aware that this is a small finding so far and that this needs to be extended in a whole new trial, most likely.
Actually, if you subtract that those 33 out of the total number of cases, you've got less than 100.
Yes.
To get you to the 70% number, and that's really borderline. So we need more data on this.
Sure.
We also need to know how long this lasts, right? Because it's only 2 months, and we need to follow people for at least a year to know, are we suddenly seeing infections, or do we need a booster? That's really important to do. So
and all these in the show where I summarized the meeting about all the trials, these are all being set up with 2-year follow-up planned. So that will presumably get at that question and also longer term issues in general.
Yeah, and I want to, agree with Alan here that in the paper they're totally transparent about all of this stuff.
Yeah, of course.
Okay, it's all there for you to see.
There's one more thing I wanted to bring up that's really, all right, duration, that was one thing. Oh, here it is. I want your opinion. So this trial or some other trial is 90% efficacy. The FDA issues an EUA. What about the control? Why are they not getting the vaccine? So do we break the trial or do we go on? The FDA hasn't decided what to do. What do you think?
That was also discussed at length yesterday.
Yeah. Mm-hmm.
Yes.
What did they decide?
And they were discussing basically that the controls would be offered vaccine sort of when their priority group was getting vaccinated, but they were going to not break the randomization. So they would call in everybody in the trial when their group came in. And if they had previously been a vaccine recipient, they would get placebo. And if they had previously been a placebo person, they would get vaccine. So they were still blinded to themselves, and they could do an analysis of sort of what happened after vaccination versus when you were still a control person. And there was even a fancy name for that kind of trial that I don't remember right now. But that was actually one part of the discussion that did go on for quite a while.
Great. Very good.
Brian, good for you watching that whole thing. How long did that go on?
I didn't watch the whole thing. It went from 9 to 6. And I did not make it all the way through. But I watched a fair amount of it.
Yeah, I got the notification about it. I was like, I'm not making it to this, but thank you.
It was a very good thing to turn on while you were doing, other work in your emails and other things. Yeah.
At 5:30, I was scheduled to do a twive with Daniel, so he's not showing up. I said, "Hey, you come?" And he goes, "Just a few more minutes. They're voting. Hold on." I said, "Yeah, I got it. Come on." Then he came on. He said, "All right." I said, "What's the result?" He said, "I'll tell you during the recording." He wouldn't tell me. All right. We have another interesting paper which has a couple of cool aspects to it that I think you will like. It's published in Nature Microbiology. Therapeutically administered ribonucleoside analog MK4482/EIDD2801 blocks SARS-CoV-2 transmission in ferrets. And 3 authors. Holy cow!
3 authors on a SARS-CoV-2 paper. Is this legal?
Is it legal? Right. Robert Cox, Joseph Wolf, and Richard Plemper, who was on twive earlier this year. I visited Georgia State last year, at the end of last year. He was on a twive with some other individuals, talked about antivirals. And so this is from his group. Very interesting. So the story is here. This drug, MK4482, we'll call it that, is a prodrug of a nucleoside analog. And I'm going to show you what this looks like because we have tech here. Here's MK4482. It's a prodrug. What does that mean? Well, these are orally delivered drugs. They're taken up and then they get modified to form the actual drug. So that this is a prodrug of N4-hydroxycytidine. Okay, let's go all the way to the right. I'm sorry for people who are just listening. I'm showing a picture of cytidine, one of the 4 bases that goes into RNA and DNA. There are no phosphates on it. It's just a ribose and the base. Right, and there's the cytosine. What is it? The base alone is cytosine. Is that right?
Yes.
And with the sugar is cytidine. Is that correct? And it would be
I believe so.
cytidine.
You just stumped everybody with a biochemistry question.
That is correct.
I think this is an important thing to know. And then cytidine triphosphate, right?
I know I had to know it for a test at some point.
This would be one of those moments where I would say, well, students, I was waiting to make sure that you knew the answer.
Yeah, that's right. That's right. Why you look it up. So cytidine has the ribose sugar, 2 hydroxyls. It's here, and then the base is up here. And you notice this has an amino group at the top, right? So the actual drug, N4-hydroxycytidine, it's exactly the same, except it's got a hydroxyl on the nitrogen. And that's it. And that's enough for it to be inhibitory in a mechanism we'll say in a moment. The prodrug has a little extra stuff on the ribose. It still has the NHOH on the cytidine, the cytosine, but it's got some extra, what would Rich call it, spinach or something on the chicken wire? Spinach. Yeah. So this is, chicken wire here, as Dixon would say. It's got some extra, and that's cleaved off when it gets into you. I don't know if it's cleaved off in the gut or in cells. It could vary.
My money's on the liver.
Liver?
Yeah.
The liver is the master chemist for all of this kind of stuff.
And this is a very, very common theme in pharmacology where the drug that you take is not the active molecule. Your body converts it into the active molecule.
The drug you take is not equal to the drug you make. Is that a song?
That's exactly right.
That was the end of the Beatles.
So you take it up, it goes into, you take it orally, which is nice here. This is an oral drug. And it's not a drug yet, it's a candidate. But then it gets in the circulation, goes to the liver, it's modified. Then this gets into cells, and in the cell it has to be triphosphorylated, cellular enzymes. Otherwise, it cannot be incorporated into RNA. And in general, molecules with phosphates don't get into cells very well unless you do funny things to them. And so this is an example of that. Okay, so that's the drug.
This drug was previously developed as an influenza inhibitor. This isn't some brand new thing that they came up with for SARS-CoV-2. But the oral bioavailability is really a very important point, because the only thing, the things that we've found that have efficacy against SARS-CoV-2 so far are all IV, or
that's exactly right. That's exactly
have other
right.
issues, which is fine if you're, a highly placed government official who gets gold standard health care and rushed to the hospital as soon as you test positive and you get injected with all the latest stuff. But if you're some ordinary schmuck like us and you don't show up to the hospital till you're coughing your lung up, then an oral drug would be better.
Or you could be a celebrity lawyer, right?
Any of those, yes. If you're in the news enough, you get a higher standard of care. But this is something that, if approved as a drug, which is still a big if, as we'll probably discuss, could be handed out much more easily.
Right. And that becomes really important with the timing issues that we've talked about before.
Yes.
We've talked about how some of the other drugs are maybe less efficacious than they could be because they're only being given to very severe patients who are already in the hospital and who are already hospitalized. If they're given IV, you kind of need to be in a hospital for them.
Right.
This is something that could be given to people perhaps earlier at home to many more patients.
Dixon, were you going to say something?
I just wanted to say what happened to the hydroxychloroquine arc that started so robustly as a central administration recommendation for being treatment, and virtually nobody in that administration ever took hydroxychloroquine as a cure.
Did y'all hear him? Did y'all hear him? Nobody in the administration recommending hydroxychloroquine ever took hydroxychloroquine. Yet our Black brothers and sisters were, the natural people, the natural immunity people of the world, were insisted that hydroxychloroquine was an aid against it. Now the science came out, said otherwise, and we backed that up with other data and trials that actually took place. But we trialed people on something that came out of the White House and nobody in the White House had ever did or experienced or anything like that. So this is pretty much a good bill, but it extends out beyond the timestamp. And what I'm gonna actually do is let it go just a little bit further, and let's see if we can get to some Q&A, because I want y'all to hear what how questions are being asked and things of that nature.
Curative. And they were so adamant about how effective it might be. I'm just,
It's dead in the water. It was trialed and it doesn't work. It doesn't work. In fact, it harmed some people.
Yeah.
Yeah, but there's still people that are touting it, though.
Yeah, but they're wrong. They should move on. Let's move on. You know what that phrase means? Move on.
Move on.
No idea.
So the IV business, even for the monoclonals, it's hard because they have to be given IV. But now they're making infusion centers outside of hospitals to try and deal with that. You can go in for 2 hours and get an infusion, and you have to be in a certain risk group. So you have a positive test and you have a certain age, comorbidity, then they'll send you to get monoclonals, and they're hoping that helps because it's a little earlier than being in the hospital, right?
That'll probably help a few people. And again, if you're in a position where you can just rush out and get this kind of treatment, then lucky for you. But but a pill is what you want, because then great. You could call up your primary care doctor, say, hey, I just tested positive for COVID-19. They say, okay, I'm writing you a script, go to CVS, pick it up.
Yeah, it's like Tamiflu right now, right? Or Lenza.
Yeah, exactly.
You get a script, you take it. So this drug, as you'll see, is really good for ferrets. And
yes.
As we know, ferrets are not humans, so that has to be tested in people. But ferrets, and here's one of the things I wanted to point out. So they infect ferrets intranasally. And ferret is a mustelid, which includes mink. So we know a weasel genus.
Or a stoat.
Oh, here we go with the stoat again. Didn't we go through the stoats before?
Are we going to start a stoat arc?
I already forgot what a stoat is. It's fine. But anyway, they put the virus intranasally into ferrets. They use 2 doses. So, I keep getting this nonsense with dose. Every time I interact with people, they're always like, doesn't dose make it more severe? And no. And I'm going to show you these data. And, the other day I looked through all the animal data, and most of the time they don't use multiple doses, especially in non-human primates, because it's pretty expensive. But from what I see, you can give more doses. They get a higher fraction of animals will get infected, but they get the same disease. So here they give 1 × 10⁴, 10,000 or 100,000 PFU intranasally into ferrets. And the ferrets get infected. They can measure that by either doing nasal wash and doing plaque assays. Yay, they do plaque assays. And right after infection, in the group that get 100,000 PFU, the virus in the nasal wash peaks at about day 3. And then by day 7, it's gone. And by day 7, that's another song. By day 7, that's the by noon he was gone. That's the poem by John Updike, right? So you get to speak, and then if you give them 10,000 PFU, there's a little blip at 4 days, but they're basically not infected. So that's the threshold. 10 to the 4 is not enough to infect a ferret.
That's a pile of virus.
It's a lot of virus, right? Infectious virus.
10,000. 10 to the 5th
It's
is
a lot.
100,000.
And they're dripping it right in the nose of the little guys,
Yeah.
right? It's not like it's moving around in the air on droplets. It's putting right in there, and it's not enough. Now, their susceptibility may be different from humans, but
one of the things that does happen with ferrets is that they can spread it between them.
they can. And
So
that's what
while
they
it
did.
does require this huge inoculum to get the train rolling, once one ferret has got it, you can put it into a cage with
another.
uninfected
Yes.
carrots,
Carrots,
uninfected ferrets, and
maybe they give them
it'll
carrots.
spread.
Yeah.
Well, right. I don't think we've seen it quite that promiscuous.
That would be impressive.
Yes, I would be amazed. But you could put it in a cage with uninfected ferrets, and they'll spread
transmit it. Now the thing is, this curve, 7 days. So then they say, what if we give this drug to the ferret? So they can give it to them orally. They have to put a tube in and put it in because they don't have pills. The ferrets won't probably take a pill.
I think from my experience trying to put pills in cats, I can imagine a tube would be very helpful. Yeah.
So they treat them 12 or 36 hours after they've infected them, and then twice a day for 3.5 days afterwards. Okay, so there's a lot. It's twice a day, 3.5 days, and that basically gets rid of the infection. There's a little bit of RNA that sticks around for a while, but no infectious virus. All right. And of course,
I liked that they looked at that, because you do see the RNA sticking around.
It does, yes, but it's not infectious.
And you do a plaque assay, and there's nothing. The virus is not infectious, so the RNA is
All right, so let's skip ahead a little bit.
an after drug for the party.
But right,
There's no virus to transmit.
yes, which they don't do to a.
Sure.
I think this. So by the way, the virus introduced
Right, 9
It's almost
years apparently.
years.
yeah.
Okay. All right. Time to do some email. I have to take this first one because this was.
All right, I'm gonna play the live on this one. I want y'all to look at their facial reactions to some of the stuff that's asked, and I think it's important that we see it.
Do this. I can show you all the grants I wrote about this kind of thing and couldn't get funded. So it's really unfortunate.
Because what were the odds of a bad pandemic coming along, right?
Pretty good.
Yeah.
Well, the corona's every 10 years, apparently, right?
Yeah.
So we have to keep twive going for another 10 years, see what happens.
See what the next virus is.
So this is 2030, that decade, the 2030s, right? Everybody, are you guys all in for this? You want
Yeah.
I'm there.
As a matter of fact, I'm looking forward. This is sort of previewing my pick, but I'm looking forward to a twive from the moon. Okay. So that's going to take 10 years.
Yeah.
Okay. All right. Time to do some email. I have to take this first one because this was, we talked about papain,
Today,
right?
and things only New Yorkers will understand.
So papain protease in terms of the viral genome last time, or very recently, and I got on this little spiel about how there used to be this juice bar on 86th and 3rd when I was a grad student. I used to pass it, and I go, "Wow, papaya and pineapple juice, right?" And both of them have proteases in them that help your digestion. In theory, I guess that's what it is. So Andy, who's an MD at the FDA, okay, he's involved in cardiovascular devices, a loyal podcast listener. I love it. 86th Street and 3rd Avenue, where peptidase meets thirst. And he sends a picture of Papaya King. I forgot the name. And there's a link to a blog post, an underappreciated place in New York, the spot where allegedly the papaya drink was born, the corner of 86th and 3rd. Signs touted papaya's superlative health benefits, nature's own revitalizer, vitamin-packed health giving. And Andrew writes, alas, randomized trials are lacking. Thank you. It's great. That's it. That's exactly how I remembered it.
Yeah.
And if you go,
and there's a New York hot dog chain, famous New York hot dog chain called Gray's Papaya.
Correct.
Which was started by one of the people who founded Papaya King and left that to start Gray's Papaya, which serves a competing papaya drink. So, as well as hot dogs.
They have pictures on this website, and it's a really narrow, it's basically the counter and then 6 feet or so of space. Yeah. Everybody's crowded in there. And of course,
it's basically a food truck that's been installed in the wall.
I never went
in.
Still
there, though. It's still there.
Yeah.
I don't know. You guys may not have ever ended up on the east side.
A couple of times.
But
I was at Rockefeller for 3 years. Come on.
And that's what 86, this is actually, what is it, Germantown. Wonderful Gray's German restaurant.
Is I think it's on Broadway.
So you could go eat.
Gray's is on, they're in several locations, including West Side, but this Papaya King exactly. is on the east side.
But, you could go to, there were some great German Bavarian restaurants in this area.
It's
you
true.
Could go and eat way too much heavy meats, and then you could go get a papaya drink and digest it.
And there's Kinsale's is right in that area, isn't it?
Kinsale's.
The bar that's got.
I think so. Yeah, I think so.
They've got a phone on the wall that's actually an internal line for is it Mount Sinai or Cornell that they're closer to?
Mount Sinai. They would be near Mount Sinai.
They're at Mount Sinai. Yeah. So they've got they're like the hangout for people who work at Mount Sinai, to the point that they wired an internal phone there. So if people, doctors got paged, they could pick up the line.
Yeah, there it is. It's 3rd Avenue and 93rd, 94th.
93rd.
Which is where I used to live, actually. I lived on 94th between 3rd and Lex. But it's closed. Yes, I remember Kinsale. It was right around the corner from us. Yeah. Yeah, so this brought back memories. That's why I'm kind of dwelling on it. Nostalgia, yeah. I think nostalgia is good from time to time.
As opposed to myalgia, which is not good.
No. And arthralgia, also not good.
That's also very bad.
Alan, can you take that next one?
Sure. Neil writes, we've seen an enormous number of infections at pork processing plants, slaughterhouses. And while we have seen cats, mink, and even tigers test positive for SARS-CoV-2, aren't pigs' ACE2 receptors nearly identical to humans? And as Vincent aptly points out with a reference, receptors are not the only determinants of infection. I would also add that receptors and infection of the livestock are not the only determinants of spread in a slaughterhouse, or even the primary determinants. The reason we see this in meatpacking plants is that this is a work environment that has people shoulder to shoulder in a cold, wet space for long periods of time. And these are people who generally do not have access to much healthcare or testing or really anything else, because in many cases they're undocumented workers or other folks at the bottom of the economy. That's why we've seen outbreaks from slaughterhouses and meat processing plants, and it's not about the pigs.
The paper I linked to shows that pigs are not very susceptible to infection. So it doesn't matter that the ACE2 is nearly identical. Once the virus gets in, there are other determinants, and pigs are not there.
Yeah, no, the problem is the people.
Good thing.
The problem, and not necessarily the people who are catching it. The problem is the social structure that's put them in that situation.
Brianne, can you take the next one?
Sure. David, who's an MD, writes, to my new friends at twive, you have helped me live and experience a smart life every week during this pandemic. So lucky am I, and lots of others, to hear such intelligent discussion. Has this medium made all of us somehow closer? I do think that this is true. I am just a cancer doc, who rarely thought about virus until the last months. I still don't quite understand the endemic EBV, HPV, and VZV, and probably many others. I knew that I needed to learn more about virus, and then I found twive. Lots of comfort in hearing thoughtfulness from all of you. Very nice to be learning lots again. So smart and inquisitive you are. Vincent is the leader. He is a new day Socrates, and with his questions, I hear his kind style. He brings out the best in all. Brienne, the youngest of the group, a smile so sweet, and with deep interest, insight, and spirit, drew.edu is a rhyme in itself. Rich, no need to smile, never dour or sour, lots of experience and perception of what really matters. He understands that there is a difference between what we can do versus what we should do. Kathy is sometimes quiet, but when she speaks, her ideas are profound. Each moment that I listen, I wait for her insight. So here I am in Central Massachusetts, new virtual friends and colleagues. I am very thankful for your twive project. We will survive. I'm really sad that I didn't get to say nice things about Alan or Dixon.
So, Dixon, do you want to read your addendum and then I'll read mine?
I'll read mine. I said, I guess I'm chopped liver. That thought makes me shiver.
Good.
And I would add, I'm also left out. How crass. Central once again forgets Western Mass.
That's good. That's very good. They belong with the poem.
Yes.
Good. That's pretty good. Rich, can you take the next one?
Dora writes, I signed up some time ago for the Novavax vaccine trial. Novavax vaccine trial. Today they called and screened me. The call center agent had no answers to a single one of my questions and said they'd be answered at my first visit, for which I'd allegedly be paid $150, even if the answers to my questions led me to immediately walk out the door. I didn't even know whether to believe it. It turned out they couldn't have answered my questions. One question involved where I'd be tested for COVID if I became sick and where I'd be expected to get treatment. I never got any answer at all to this, not even we don't have the answer. This is an obvious confounding factor. If they need to diagnose it, then I may need to ride my bike 8 miles in the winter with a high fever or trouble breathing. If they go by my doctor's diagnosis, my doctor couldn't diagnose a boil on his butt. If it happens to test positive by the time the doctor's office and the lab mess it up, cowabunga. Now I ask, what if you don't know if you've already had the coronavirus, since I don't, and it only matters if you've ever had a positive test? I think that I and most people I know had COVID in March. Second, and most important, the trial is for 5 years. A vaccine should be available to high-risk people like myself in 2 to 3 months. I asked if they'd give everyone a shot at that point, or all high-risk people a shot at that point, or even tell us if we actually got the vaccine at that point. I pointed out that not only would I go get the vaccine if I hadn't already had it, or couldn't determine if I already had it, but so would many people. I was told that we are free to leave the study whenever we want. After 2 or 3 months, they've not learned anything yet. I also pointed out in my email to them that less scrupulous people will get the vaccine elsewhere and not tell them. They'd gain the most valid information by vaccinating all of us at that point and then following us. If a third of people who were vaccinated subsequently get COVID, they'll know it works about as well as a flu vaccine. I was told they have no idea. In my mind, this must mean this study is not being well or ethically run. For certain, any results after the date when people get the vaccine, if they want to, won't mean a whole lot. Yours, Dora. So it sounds like Dora did not have a very happy experience with the vaccine trial. I don't fault at all the call center people for not answering questions. They're just taking calls. Sounds like she could have gotten maybe some better answers from the people running the trial. However, I mean, here we even stumbled, and apparently in the FDA conversation about the vaccines, there was a lot of conversation about what do you do with people who are in the trial, and it's a complicated question.
Her question about how she's going to get tested if she has symptoms, but I think we saw in the Lancet paper we did that you have a self-test kit. They'll send you swab yourself, you mail it in, so you don't have to ride your bike 8 miles in the cold. So at least there's that, Dora.
Yeah, I think this is a mix of questions that that had easy answers, but she didn't get referred to the right person.
Yeah.
Because I'm sure I know Novavax is setting this up, according to standards, and I'm sure the answers to this are available. And I think her email just got sent to somebody low level who blew her off. But that last question about what do you do when the vaccine comes out, the speculative question, as Rich said, that's something where there aren't good answers.
Although it depends, like as we heard from Brianne, they discussed yesterday that they would offer the vaccine when your time comes up to be vaccinated.
Yes.
And
I'm sorry. Yeah, there are good answers. They're not necessarily
great
obvious or satisfying or definitive, and they are subject to some debate.
That's right.
Yeah.
Well, I'm not surprised. With so many trials going on, things like this are going to happen.
Sure.
Sorry about that. Dixon, can you take the next one?
Anthony writes, hello, Twiv. Shouldn't Dr. Fauci be advocating for a recovery-style randomized study to assess head-to-head the efficacy of Pfizer and Moderna vaccines and any others? If not, how come? Wouldn't such a randomized study be advantageous, especially for medical frontline workers and in persons and in nursing homes at this point in time? And then Vincent adds, see, mix and match Pfizer and Oxford jabs, the London Times, and it's got a big story about that. So are any of those comments valid, Vincent?
Well, somebody's in trouble. I can hear the sirens. Is that you?
I can hear the sirens in your background too, Vincent.
No. This is Santa Claus. Are you guys kidding? This is Santa Claus.
It's Dixon. It's Fort Lee. I'm too high up here. So this Times story is they're going to put both Oxford and Pfizer vaccines. They're going to do mixing and matching to see if combining them makes a stronger immune response. They're not going to pit them against each other, but I think that's an interesting approach. And the other. Didn't you say that the Sanofi one said they would try them?
Sanofi is now pitching the idea of putting theirs in a comparative trial, which is a gutsy move, right?
Yeah,
I mean, if they end up having, so they're pretty confident in their vaccine, which they should be. It's using an established tech. So I think we are going to see these types of trials. The priority was on getting through initial trials with the simplest design possible, because you don't want to compound it. I mean, imagine if we didn't have a phase 3 answer for either Pfizer or Moderna, we had a head-to-head trial between the two, and we had to deconvolute all of that before. No, you do these trials first individually. And now, as more vaccines come online, I think there's going to be more and more pressure to do these comparative trials. Are you as good as what we've already got? So yeah, that is definitely going to be in the works.
I don't understand the reference in this to a recovery style randomized study. Can you enlighten me? Is that a particular type of study?
Yeah, all these clinical trials have cute acronyms. So
the recovery trial was a specific trial designed for COVID-19 to test many different things at once. Right. And it was very flexible. You could add and take away treatments. Like it looked at dexamethasone, tocilizumab, convalescent plasma, monoclonal antibodies. So very flexible, this very clever and flexible trial design where you could add and take away things along the way. So and you can compare things. Yeah. So
which is appropriate for it. Yeah, that's appropriate for a trial where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other kinds of tests, right? You don't want to be doing that initially.
Yeah, so you want to sign up 30,000 people for a trial, and you want to ask one question, and then get that answer, and then later on down the line, you can sort out which is the best vaccine.
Right. And then you want to have something out there. And you can decide which of the things that are out there are the best later. But right now, we need to help some people.
I mean, in retrospect, it seems brilliant now, right? They did this mRNA vaccine and it worked. They could do it quickly. And we were very skeptical at the beginning. We didn't know, right? And this focus on spike turned out to be okay.
They didn't know either.
No, they had
to be honest. And it's easy to confuse luck with brains. I mean, a lot of very smart people worked on this, but let's not downplay the role of.
What are you talking? I swear you can't hear.
Where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other kinds of tests, right? You don't want to be doing that initially.
Yeah, so you want to sign up 30,000 people for a trial, and you want to ask one question, and then get that answer, and then later on down the line, you can sort out which is the best vaccine.
Right, and then you want to have something out there, and you can decide which of the things that are out there are the best later. But right now, we need to help some people.
I mean, in retrospect, it seems brilliant now, right? They did this mRNA vaccine and it worked. They could do it quickly. And we were very skeptical at the beginning. We didn't know, right? And this focus on spike turned out to be okay.
They didn't know either.
No, they had to be honest.
And it's easy to confuse luck with brains. I mean, a lot of very smart people worked on this, but let's not downplay the role of a little bit of luck in there. We didn't know going into this was going to. But
a lot of technology that had been developed before.
Yes.
By the way, this idea of comparing vaccines reminds me of the polio vaccine.
Individually. And now, as more vaccines come online, I think there's going to be more and more pressure to do these comparative trials. Are you as good as what we've already got? That is definitely going to be in the works.
I don't understand the reference in this to a recovery style randomized study. Can you enlighten me? A particular type of study.
Yeah.
All these clinical trials have cute acronyms. So
the recovery trial was a specific trial designed for COVID-19 to test many different things at once, right? And it was very flexible. You could add and take away treatments. Like it looked at dexamethasone, tocilizumab, convalescent plasma, monoclonal antibodies. So very flexible, this very clever and flexible trial design where you could add and take away things along the way. So and you can compare things. Yeah. So
which is appropriate for it. Yeah, that's appropriate for a trial where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other kinds of tests, right? You don't want to be doing that initially.
Yeah, so you want
frontline people and all people. Are y'all okay? Let me continue.
But you want to sign up 30,000 people for a trial, and you want to ask one question, and then get that answer, and then later on down the line, you can sort out which is the best vaccine.
Right, and then you want to have something out there, and you can decide which of the things that are out there are the best later. But right now, we need to help some people.
I mean, in retrospect, it seems brilliant now, right? They did this mRNA vaccine and it worked. They could do it quickly. And we were very skeptical at the beginning. We didn't know, right? And this focus on spike turned out to be okay.
They didn't know either.
No, they had to be honest.
And it's easy to confuse luck with brains. I mean, a lot of very smart people worked on this, but let's not downplay the role of a little bit of luck in there. We didn't know going into this was going to. But
a lot of technology that had been developed before.
Yes.
By the way, this idea of comparing vaccines reminds me of the polio vaccines. There were two oral poliovirus vaccines developed, one by Albert Sabin and one by Hilary Kaprowski in the 50s. And so they said, okay, which one are we going to use? So they did a match off of the two. They did a trial where they compared the two. This was run by Joe Melnick at Baylor. And Sabin vaccines slightly ahead of the Kaprowski. So those are the ones that we're using. And Kaprowski was always very upset about that. He said, no, mine should have been used.
In retrospect, has there been any comparison? Was Kaprowski's virus kept around?
Yeah,
and, you know, because since that time, we've learned that there are issues with the oral vaccine, in particular shedding revertance to neurovirulence. And I wonder if Kaprowski's vaccine did the same thing. Maybe in the long run, it would have been a better deal.
Yes, it has been looked at, and I don't remember the results, but that has been examined. And I don't think the same attenuating mutations are present in the Kaprowski vaccines. So I can check. I will look. That's an interesting point. Now, of course, the Kaprowski vaccines were trialed in Africa, and he was blamed for, introducing HIV.
That silly book, yes.
It was silly, which is real wrong, but they said he grew up his virus in chimpanzee kidney cells, which he got from Africa, which had, SIV. It's just crazy theory, but it's not true. It's not true.
So, Vincent.
All right, do I need to do this again for you? I done rewinded it 3 times, and he ain't even talking about no people. He ain't even talking about black people. God damn, I don't understand how you. I'm sitting here. I done listened to this 5 times. This show, I done listened to it before I even came on air with this. I did this yesterday too. I don't understand it. So let's go back. God damn, listen.
Albert Sabin and one by Hillary Kaprowski. So Spike turned out to be
they
okay.
Didn't know either. For a trial, and you want to ask one question.
Monoclonal antibody.
Was on getting through initial trials with the simplest design possible, because you don't want to compound it. I mean, imagine if we didn't have a phase 3 answer for either Pfizer or Moderna, we had a head-to-head trial between the two, and we had to deconvolute all of that before. No, you do these trials first individually. And now, as more vaccines come online, I think there's going to be more and more pressure to do these comparative trials. Are you as good as what we've already got? So yeah, that is definitely going to be in the works.
I don't understand the reference in this to a recovery style randomized study. Can you enlighten me? A particular type of study.
Yeah, all these clinical trials have cute acronyms. So
the recovery trial was a specific trial.
I see.
Designed for COVID-19 to test many different things at once, right? And it was very flexible. You could add and take away treatments. Like it looked at dexamethasone, tocilizumab, convalescent plasma, monoclonal antibodies. So very flexible, this very clever and flexible trial design where you could add and take away things along the way. So and you can compare things. Yeah. So
which is appropriate for it. Yeah, that's appropriate for a trial where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other kinds of tests, right? You don't want to be doing. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people. And
for frontline people and all people. Frontline people and all people. One more time for the slow poke. This is why you don't deal with, you need to stop hanging out with Zion Lex and Jeremiah Judah.
To check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later do the other.
Now that we have slowed it, don't try to tell me to shut up. Don't try to tell me to shut up. The man said frontline people and all people. How you get black out of that? Frontline people and all people. What are you saying? You heard black one more time, yo. Am I tripping? Mika, am I tripping?
The kinds of tests, right?
where you've got a whole bunch of already approved drugs.
These were approved drugs that they wanted to check for COVID. It's the recoverytrial.net. You could see more about that there. I think the other consideration, Alan, is that we wanted to get something out there fast for frontline people and old people, and then later.
All right, I ain't doing it no more. I ain't doing it no more. All right, yo. So that's gonna end our 12 talk right there, man, because they just gonna ramble on for the next 40 minutes. Even though it's good information, a good bill, we actually got through a lot of the video. We did skip ahead and get to the Q&A, and we did get into a lot of things that we actually dropped in the source. We realized that Sanofi didn't make their deadline to be able to do what they need to do. So instead of them wasting their research, they're going to come back in 2021 and do a do an actual study with all 5 vaccines, and we could look at the efficacy and we could look at the data regarding that. That was pretty good. Here I gave you the job opportunity. I'm gonna put it back in the chat again. I don't know who watches these shows and who may be qualified for this particular job, but there is a job related to what's going on out there, and they are looking for people who are qualified for this. So anytime they're looking for things that's going on, I think that our people should be aware of that. Share it with a family, friend, or member, somebody that may qualify for this stuff, and we'll do it from that perspective. On to the meat and potatoes. Before we wrap up and end the show, in the description of the video, I put all of these last articles, 5 FDA articles regarding the committee's meeting on December 10th, and so that you can be abreast to what's going on, what happened in the trials, how the vote took place, how did we come to this particular perspective. You know what I'm saying? So we're gonna run down real quick, and I don't want to really get into all of the data, but I gave it to you in case you wanted to read about it and then come back to it one day and says, hey, Shawn, what did you think about page 46 on the such and such? You know what I'm saying? And then we can actually deal with it from there. I did want to come down to something that I felt that was very important to not leave off the show, and that was the benefits risk assessment in context of proposed indication and use under the EUA. Known benefits. The known benefits among recipients of the proposed vaccine related to the placebo are reduction in the risk of confirmed COVID-19 occurring at least 7 days after dose 2, reduction in the risk of confirmed COVID-19 after dose 1 and before dose 2, reduction in the risk of confirmed severe COVID-19 anytime after dose 1. The protocol specified 2-dose vaccination regimen was highly effective in preventing PCR confirmed COVID-19 occurring at least 7 days after completion of the vaccine regime. Additionally, primary efficacy analysis in all available efficacy population, including participants who controlled the protocol violation, show consistency with outcomes in the primary analysis population. Our efficacy findings were also consistent across various subgroups, including racial and ethnic minorities, participants aged 65 years and older, and those with one or more of the following conditions: obesity, diabetes, hypertension, and chronic cardiopulmonary disease. While limited available data suggests that individuals with previous SARS-CoV-2 infection can be at risk of COVID-19 reinfection and may benefit from vaccination. So I'm gonna read that last line again because people are hung up on this. And you also heard in the video that they told you you're not going to gain immunity from getting the virus. All right. So while limited available data suggests that individuals with previous SARS-CoV-2 infection can be at risk of COVID-19 reinfection and may benefit from vaccination. Among participants with no evidence of COVID-19 prior to vaccination, the vaccine was effective in reducing the risk of COVID-19 and severe COVID-19 after dose 1. Fewer severe cases were also observed in the vaccine recipients relative to recipients of placebo during the follow-up period after dose 1. The findings post-dose 1 from post-hoc analysis cannot be because the period of observation is limited by the fact that most participants received the second dose 3 weeks after the first one. Unknown benefits and data gap. Duration of protection. As the interim and final analysis have limited length of follow-up, it is not possible to assess sustained efficacy over a period longer than 2 months. All right, so we're looking at an efficacy period as of right now. Yeah, my bad. I'm probably draining y'all out. You know what I'm saying? I know that might be a little bit more important. So let me hurry up. Or you know what I'm saying? I'll do this for the people that'll rewatch the show. And then you know, I know y'all might want to attend that. You know what I'm saying. But as the interim and final analysis have limited the length of follow-up, it is not possible to assess sustained efficacy over a period longer than 2 months. Sosa, I know you may be watching. So right now we're dealing with a too much efficacy plot. All right. Now, goes beyond that, the more data that we get. So we're going to be a part of learning the long-term effects in our lifetime before we transition, or as long as we have breath in our body. All right. Efficacy in certain populations at high risk of severe COVID-19, although the proportion of participants at high risk of severe COVID-19 is adequate for the overall evaluation of safety and available follow-up period, the subset of certain groups such as immune-compromised individuals like HIV and AIDS is too small to evaluate efficacy outcomes. All right. Now, effectiveness in individuals previously infected. The primary endpoint was evaluated in individuals without prior evidence of COVID-19 disease, and very few cases of confirmed COVID-19 occurred among participants with evidence of infection prior to vaccination. All right. Although more cases occurred in the placebo group compared with the vaccine group. That's good to know. Therefore, available data are insufficient to make conclusions about the benefit in individuals with prior SARS-CoV-2 infection. However, available data, while limited, do suggest that previously infected individuals can be at risk of COVID-19 reinfection and could benefit from vaccination. They timed that up too on us. Now, I'm not going to get into all the other things. This is why I'm dropping it off how I'm dropping it off. And then here we go, known risk. Right? So we all know about the known risk. So let's get into the systemic adverse reactions compared to the placebo arm. Usually lasting a few days. This usually lasts a few days. The most common solicited adverse reaction with an injection site reactions is 84%. We all know that. Coming into the game, we know there's a reaction at the injection site no matter the vaccine. Okay? It's just, straight like that. Fatigue, headache, muscle pain, chills, joint pain, fever. Adverse reactions characterized as reactogenicity, we know about that, were generally mild to moderate. The number of subjects reporting hypersensitivity related adverse events was numerically higher in the vaccine group compared to the placebo group. All right, so severe adverse reactions occurred in a 0.6% participants were among frequent after dose 2 than after dose 1, and were generally less frequent in older adults. And compared to younger participants, among reported unsolicited adverse events, lymphadenopathy occurred much more frequently in vaccine group than the placebo group, and is plausible related to vaccination. All right. Now, serious adverse events, while uncommon, represent medical events that occur in the general pop at similar frequency as observed in the study. 3 SAEs in the BNT group were considered related by the investigator, but not the sponsor, as a related study vaccination. Shoulder injury, ventricular arrhythmia in a participant with known cardiac conditions, and lymphadenopathy, temporarily related following vaccinations. We consider 2 other events as possible related to vaccine. The shoulder injury possible due to vaccine administration of the vaccine itself, and lymphadenopathy, however you say that word. Let's be more specific, because I don't mess with this word. It's new to me. Was largely associated with biologically plausible. Boom. Really? Yo. I guess because it's on a PDF. Refers to an enlargement of one or more lymph nodes. All right, okay. All right. Being shaped glands in the neck, armpits, chest, groin, and abdomen. Gotcha. Okay. Boom. All right, now I know what you're talking about. All right, cool. Y'all see that? I see that. Yeah. I used to feel this little thing in my neck all the time. This little, yeah, okay, under the armpit too. Okay, all right, we cool. I know what they're talking about. All right. No specific safety concerns were identified in the subgroup analysis by age, race, ethnicity, medical comorbidities, or prior SARS-CoV-2 infection, although participants 16 to 17 years of age were enrolled in the phase 3. Safety data in this age group was limited. However, available data are consistent with the safety profile in the adult population and its biological reasoning and extrapolate the greater safety experience in adults, in particular younger adults, to oldest pediatric age group 16 to 17. All right, this is why I'm giving you the homework. I put all of them in there. Yo, do your homework, man. You know what I mean? Do your homework. Make sure you make an informed decision. By the time 6 months is up, we can revisit any conversation along the way and we can get more into it and we can deal with everything that we need to deal with regarding this. I'm not going to hold you up any longer. I expect everybody to drop off. MBK is live beating up elbows. Like the show, share the show, tell a friend, tell a friend. Stay tuned to Kofi Pie Side TV later this afternoon. And later on this afternoon, there's going to be a nice, some nice information by the team. We'll get into that. You know what I'm saying? No, Mika, we're not doing no after show. You know, MBK has class in session. I'd like to appreciate everybody for hanging out with me the past 2 hours and 15 minutes. Thank you again. This is Science with Shawn. I don't know the next time I'll be on to do Science with Shawn, but hopefully it'll be some more good news. Maybe we'll revisit some of this information from the FDA and kind of dig into their files or whatnot. But again, all the information in the description. Go to the merch store, support the merch store. Spiritual Combat is out now. Killmonger's Corner Volume 2 is out now. Support our latest works, man. Please. We don't do GoFundMe's and we're not trying to beg anybody for money. We're trying to put out sound scholarship and merch that that you guys respect and love. You know what I'm saying? We don't have a big following. We're not here to please everybody. Some say we're boring. They give us all kind of names. So I appreciate everybody hanging on. We've been hanging on strong to a bunch of people who haven't been a part of the chat room conversation. But hopefully from this bill, everybody gains some type of knowledge out of the situation and can use this and reapply it, or even come back to these videos, Science with Shawn videos, and share them with your friends. Let's see if they can learn something too. Our job is to help everybody make an informed decision regarding the COVID-19 vaccine, the mRNA vaccine. On that note, Shimon Hotep and ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. after show on the way. Peace.